talker for today, I think we can get started.
All right, thank you, Megan. And I'd like to thank everybody for joining us today for the Trent Biology Department Seminar Series. As this seminar series has moved to a virtual setting, we are now able to expand our audience beyond the faculty, staff, and students here on Trent's campus. And this week, I'd like to extend a special welcome to Trent Biology alumni, who I understand are some of whom are now in attendance and joining us. It is my pleasure to introduce our seminar speaker this week, Dr. Dedmer van der Waal, who is joining us live from the Netherlands. Dr. van der Waal received his PhD from the University of Amsterdam in 2010, where he worked on aquatic ecology, ecological stoichiometry, and harmful algal blooms, which I think some combination of that we're going to hear about today. He is now a senior scientist at the Netherlands Institute of Ecology, and his talk will be on global change and harmful algal blooms in marine systems. Welcome, Dedmer, and the floor is all yours.
Thanks, Paul. I'm just checking if I'm not muted. No, all looks good. Right, so thanks a lot, Paul, for well, having me over. It's a bit weird, of course. I'm on the screen there, sitting in my own home office here. But it's great. It's great seeing you. And well, as we also just discussed shortly, these are some of the minor benefits we may have from these times, is that we can share talks around the world, right? So today I want to talk about global change impacts on marine harmful algal blooms. And before starting, I just want to thank some of the collaborators in this work. My former PhD student, Karen Brandenburg, who is now a postdoc at the University of Utrecht in the Netherlands. So some of her results I'll be showing today. And also I'll be showing some of the large musicosm study we did quite some time ago, but it got published last year. And I also want to share some of that with you. And of course, there were a lot of people involved in that. This is really a team effort. And yeah, the person that led that part was Silke Wolrap, so also special thanks to her. Before I also continue with the rest of the talk, I also I reflected a bit on my last visit. Well, last time I saw you, Paul, was well, I visited you, and that was in 2015 when I was at Trent University for the conference on biological stoichiometry. And I recall then that whenever I visit a conference, I try to look up some artists that either are local or come from that area. And I came across Rob Gonsalves. He's from Toronto, and I'm a big fan of Escher, and that's a Dutch artist from quite a while ago. And the work by Gonsalves is very much inspired by Escher, and I think he really brought it to a next level. And I really like these kind of, well, surrealistic, dreamy paintings that he makes. And it is also something that I recognized here, being partly limnologist. So a lot of my work actually was in freshwater systems, or is still in freshwater systems. So my PhD was also in freshwater cyanobacteria. But also I worked then on marine systems. I moved to my postdoc, I moved to the Marine Institute. So I really see that well, from this well, laying down and reflecting on Earth, I look at Earth, and well, what do I see? Well, I clearly see the oceans, right? And being from where I am, and of course, if I look at the ocean, well, this is then the North Sea, just for your orientation, and I of course included it also to show a little bit where I am now. So this is the North Sea, this is the Netherlands. This tiny country here. So I'm at this moment talking from this place here. It's near Wageningen. That's where our institute is. And well, for your orientation, this is Denmark, here's Sweden. Later I will be talking about an experiment we did just right, almost just not on the map there. I will have another map for that. But of course, if we look at this water, then what jumps in I guess most of your eyes is here, these clouds here, these in the water, these are coccolithophore blooms, and that's a calcareous algae. And I'm showing this because algae play, of course, a very, very important role in aquatic systems. as most of you, I assume, know, they are of course the base of the food web, right? They convert, like all primer producers, the inorganic compounds into organic matter. So they really form the essential link between the non-living and living world. And the other aspect, especially in marine algae, is that they also play a very important role in biogeochemical cycles. So with the vast surface of Earth consisting of oceans, oceanic primer production really plays a key role in transporting carbon, for instance, from the upper mixed layer down into the ocean interior, and that's called the carbon pump. And in that process, part of that material is of course remineralized, but also part is really stored down deep, and with that removed from the atmosphere. So they really are very important players in the ocean carbon and also elemental cycles. And algal blooms of course play a key role, because if they are really periods of high of proliferations of certain species to really high densities, and of course, yeah, that you can then see from space, as we see here, but also across the Atlantic, which is more in your direction. So I think this is Newfoundland, or I'm not sure if I say it correctly, Newfoundland or something like that. I've been once to an ASLO conference in St. John's. I think it's below, he's somewhere below the clouds here. Anyway, but also here we see again these clouds in the water of coccolithophores, this calcareous algae, and also here we see some green stuff. That's all I can say now. I from space we cannot see what is there really, but I would guess some diatoms are blooming there in that system. And of course they really play their roles there. Then in some cases, these algae can also consist of other species that can be toxic or a big nuisance. And we refer sometimes also to them as red tides. So this is again in the Netherlands. And I just show this picture because, I mean, this is clearly where the name is derived from, red tide. But this is not a toxic species. This is Noctiluca, which is a luminescent dinoflagellate that can, well, in nighttime, you can see very nice bright light coming from that. But this causes a lot of issues in the water because it is associated to certain toxic species, but also to skin irritation, for instance. And of course, if you have massive blooms bigger than this, I mean, this is still relatively small, then it can, of course, also cause the algae can sink to the sediment and cause, of course, sediment anoxia. Also in Canada, you have some of these red tide species. So also this is Noctiluca, again, not toxic species. But I also looked up, and obviously there are also harmful algal blooms, especially at the Pacific coast. So here we see an image of a warning sign not to eat shellfish, because these algae, they are microscopic, right? They are maybe like generally these harmful algal blooms in marine systems, they are between 15 and 50 micrometers. So they produce toxins, and these toxins generally stay within the cells. And of course, that's not a lot in one cell, right? I mean, they are very small. But if you have an accumulation in one way or the other, so for instance, they all swim to the surface, or they swarm, or they aggregate. When they die off, they can aggregate and make small aggregates, and that can wash on the beaches, or they are eaten by shellfish, then of course they all get together, and these toxins can be concentrated. And that's what happens in shellfish. So there's a bioaccumulation of toxins. And that can have really impacts on us, right? The way we use the water, for instance, for harvesting shellfish. So this is, for instance, a map on areas that have been closed for shellfish harvests in 2019. And one aspect I want to highlight here is that these toxins, there's a whole range of chemical compounds, and they are clustered, they are categorized in the kind of symptoms that they cause. So for instance, in this case, it concerns certain phytoplankton species, some algal species that produce toxins that cause paralytic shellfish poisoning. And it's really in the name there, right? So it's a neurotoxic, and this is really one of the most potent neurotoxins in algae that we find. And yeah, you can really have well, a bad experience eating those shellfish if they have accumulated toxins. If we look at these PSPs, then we also see that it's really everywhere, like PSP poisoning by different algal species, but all dinoflagellates we see really around the world. And they can have really huge consequences. So, and that can also be linked to climate change, climatic changes. So, for instance, in Chile in 2016, there was really a massive bloom causing a die-off of a lot of shellfish, but actually a lot of marine life in general. So up to sea mammals, and this really affected a lot of people, major fisheries with billion-dollar economic losses. So that really affected the livelihood of a lot of people as well. Smaller fishermen were not able to actually get their income, right? So this really has huge consequences, and that scientists really linked that event, that massive bloom event, through El Nino. Besides PSP toxins, there's a whole range of others. The other one I want to point out as well, because they come back in my talk later, is amnesic shellfish poisoning. Yeah, I forgot what it means, amnesic. I think it has to do something with memory, right? So that's ASP, that's caused by domic acid. That's one of the compounds. So I mentioned that. It will also come back later in the talk. And well, one thing there, also there we see climate trends. So for instance, in 2013 up to 2015, there was a series of massive blooms along the Pacific coast of Canada and the US. And they have been associated to heat waves in that period as well. So here we see, I don't know if you see my mouse, here you see this massive coloration of the water. So it's all really thick soup of Planitia, which is a diatom. So the previous one, the PSP toxins, are produced by dinoflagellates. Domic acid is produced by diatoms. And this well, this is really a recurring event along the coast. And sometimes it really is stronger than other times. And it might be linked to climate change. Right, so everyone knows these kind of pictures, but I do show it because for phytoplankton being primary producers, of course, this has this is important. So atmospheric CO2 levels are rising, and of course that also causes an increase in CO2 in the water. And being primary producers, phytoplankton, of course, use that CO2 for photosynthesis, right? And then at the same time, we also see increases in temperature. So we get new records of hot summers almost every year. So the world is really warming, and of course that can have really impacts also on metabolic processes in all organisms, so also phytoplankton. So these are really important global environmental changes. Also, we keep using and applying fertilizer on our lands, right? So this is just an image just to illustrate is that nitrogen fertilizer consumption is really still increasing in many places around the world. Maybe there were some mitigation measures to reduce, but still it's still very high compared to halfway last century. So we're really dumping still a lot of nutrients in a lot of systems and enriching it with nutrients. And of course, phytoplankton use these nutrients for their growth. And well, that's also what we then see in blooms. So a lot of blooms are fertilized in that sense, and we see blooms occurring at coastal systems. And yeah, as I said before, these can really be toxic to us, to mammals. So when I talk about toxicity, it really is meant to toxic effects on mammals. There are also compounds that are toxic directly to the environment. These are often called allelopathic compounds. The difference is that many of these allelopathic compounds we don't know. These are not compounds toxic to us, but they are excreted by the cells and cause harm to direct environment, so to grazers, for instance. Many of the toxins I talk about are compounds that are toxic to mammals, tested in of course, for instance, mice or other mammals to check for the LD50s, for the little doses. And they are often intracellular. So these are compounds that stay within the cells of the algae, of course, unless they die, and then they may get into the water. And the toxicity of harmful algal blooms depends on different drivers, different levels of organization. So first, if we look at this bloom, for instance, and we would establish the toxicity, clearly there is a bloom. So one of the main aspects is that if you have a high biomass, then of course you have high potential for toxicity, right? I mean, without biomass, toxicity is probably very low. But it's not just the biomass, it also depends on who's there. So on the community composition, so which species are there? Because different dinoflagellate species or different phytoplankton, even different toxic phytoplankton species can differ in the types of toxins they produce. So that really also has an effect on the overall toxicity of a bloom. Furthermore, if we want to understand further this toxicity of a bloom, we can look at the population level. So here we see, for instance, a population of dinoflagellates from a Dutch creek. So that's a brackish water creek of Alexandrium. And there, if we isolate 20 different cells, we get 20 different genotypes. So they're almost the same, but have slight differences between these genotypes. And these differences can really have an important impact on, for instance, the amount of toxins that are produced, the types of toxins are produced, and in some cases, one genotype is toxic, whereas the other is not toxic at all. And of course, that matters. So they have a lot of similar traits, except maybe for their toxic compounds. So a bloom by a non-toxic genotype will not be as harmful as a bloom by a toxic genotype, obviously. Then, if we look in the toxic genotypes, we can also see variation. Because these toxic compounds, like here we see saxitoxin causing PSP syndrome, the cells can really vary the amount of toxins they produce, depending, for instance, on nutrient availability, CO2, temperature, etc. So these level of toxins in the cell also depend on the environment. Lastly, what is also important and quite interesting, I think, is that if this compound has two groups of sulfate attached to it, it becomes actually not toxic. So it changes from a toxic compound because it blocks a sodium channel in your neurons. But if it has two sulfate groups, it cannot block that channel anymore, and it becomes practically intoxic. So understanding bloom toxicity, I'm interested in understanding bloom toxicity and also the impacts of global change. I think it's important then to look at these different components and to really understand how and by what is toxicity altered through global change. And we do that in different ways. And I think this applies to many, so we can look at different levels of organization, right? So for instance, we can go in the field, like with ships for the marine system, and then we go out there, we take samples, and that is pretty nice. It's the real world, right? But it's also super complex because we don't have any control on the system. It's there, and we take samples from that and see what is there. So to understand global change, then of course that is very difficult because we need to sample, have long monitoring programs to be able to see how global change alters it, unless we have, of course, strong gradients in our sampling schemes. The other option is then to go in the field, but make enclosures. Like these are mesocosms, and I will tell more about an experiment we did with these. And we can do mesocosms out there in the ocean, and then, for instance, increase CO2 levels or add nutrients and so on. And then we take part of that complexity, we enclose it, and then can do experiments there. We can also take the water into the lab. So this is at our institute. We have different indoor mesocosms where we have more control, right? We can control the climate as well. So we can control light, temperature, etc. So we move a little bit away from the full complexity. We get more control, but of course, it's smaller volumes and you get more side effects as well. These systems that I just described, mesocosms, are very ideal systems to understand community level effects, right? Because we can really include a big part of, in these mesocosms, indoor, we can keep a lot of the zooplankton, for instance, in there. Whereas here we can even work with small fish still in there. So we can really keep part of the food web. Another level is, for instance, here in chemostats, which we developed for dinoflagellates, which are very fragile. So we had to change the way we mix the system. Normally it's with aeration. Here we used a ball in a big air bubble, and it worked pretty well actually. And these are chemostats for testing, for instance, competition between different species. So here we can already control much more and study, for instance, competitive interaction, resource competition. So we can really look at community interactions or population interactions, because we can also work with different genotypes and see if there are trade-offs, intraspecific trade-offs, and how they result in the overall success of certain genotypes over others. And then lastly, we can do batch experiments. So these are basically bottles. Either there's an inlet of air, not very clear here maybe, but there's some air going in. So we can, for instance, very easily change the CO2 concentrations and control them very nicely. Or very simple, just use LMI flask and a stopper and different incubators, and that's also possible. The advantage of these last experiments is, of course, is that they are way more simple, especially these LMRs. So we can work with a lot of different species, genotypes individually, and really get a very good grip on individual responses to a wide range of stresses, because we can have a lot of replicates as well and a lot of stresses at the same time. So basically what we can do with these kind of experiments is get a lot of understanding on a very simple system, but it's not the real world. Whereas in the real world, well, it is the real world, of course. So we really look at the real life context and the complexity, but the complexity also means that we have to give in some of the understanding, because there's happening so much that we never can capture everything. And I think the combination of approaches is therefore very powerful to really develop understanding here, test it in the field, and go back and forth. And that's what, of course, many ecologists do as well. And well, in this context, we also do that. Right, so for today, I want to highlight some, let me see, where am I? I have two screens here. Some experimental studies, and I want to focus on a community study that we did in the field. So the first is a study we did to assess the impacts of elevated CO2 levels and temperature on growth of hub species, on harmful algal bloom species. And we did some experiments ourselves, but of course there are a lot of species out there, a lot of genotypes out there. So we thought it was maybe easier then to actually look at literature and perform a meta-analysis, so that contained part of our own data, but we could also include a lot of other species and really have a look at how do hub species in general respond to elevated CO2 and warming. Well, we looked specifically at growth rate. We also studied toxins, but I won't go into that here. And what we did is we looked at literature that contained some kind of biomass indicator over time, and we took the maximum growth rate, right? Any measure of maximum growth rate we could take, and we calculated the maximum growth rate at elevated CO2 and ambient CO2 and elevated temperature and ambient temperatures. And we expected, so our starting hypothesis was that both CO2 and temperature will increase the growth rate of the harmful algal bloom species. Temperature because of higher metabolic rates. So with higher metabolic rates, species can grow faster. And then with CO2 also, because with more CO2, well, the CO2 may fuel photosynthesis, and therefore species may grow faster. Also, many algal species have carbon concentrating mechanisms. And by down-regulating these CCMs, these so-called CCMs, they may actually gain energy and use that for growth as well. And I just want to show a little bit on that, because it's one of my other hobbies, and I thought maybe that's interesting for here as well. So what we see here is the response ratio of the K-half for photosynthesis. So K-half, maybe you remember, that's the half saturation concentration for a process. So K-half in this case means the concentration of inorganic carbon where photosynthesis is half of the maximum, right? So if you increase K-half, then you need more inorganic carbon to operate at half of the photosynthesis, which means that your affinity is decreasing. In other words, K-half is a kind of a proxy for affinity. It's the inverse of an affinity, so to say. So the higher the K-half, the lower the affinity. What we did here is we looked also in the meta-analysis at a response ratio towards CO2 over ambient of this K-half in different phytoplankton groups. So cyanobacteria, dinoflagellates, haptophytes, so the calcareous algae, for instance, are these haptophytes, Emiliania huxley, for instance, and diatoms. And what we see here is that the K-half in response to CO2 in both cyanobacteria and dinoflagellates increased, which basically means that their affinity for inorganic carbon decreased, which makes sense because they get more CO2. They don't need this very high affinity. And this indicates that they downregulated their CCMs. And the interesting thing is that we particularly see this in cyanobacteria and dinoflagellates and not in diatoms and haptophytes. And the reason for that is that these species need very effective CCMs in the first place because they have very poor ribiscos. And ribisco is this primary enzyme that is involved in, that is responsible for carbon fixation, right? But it has a very low affinity for CO2. So it's like a very poor engine in a car, but yeah, it's the only car that's there. So they have to deal with that engine, right? They have to deal with that ribisco. And that's why they develop CCMs, because CCMs, like you can think of active uptake of CO2 or bicarbonate, or the prevention of losing CO2 out of the cell, so preventing leakage. These are CCMs. And especially these groups with very ineffective ribiscos, they really need these CCMs. But then if you give them more CO2, they are the ones that can also downregulate the CCMs. And with that, maybe gain energy or elements to invest in growth. This is a little bit a sidetrack, but it's just one of my other hobbies, and I thought it was interesting to also highlight here. So these groups might be particularly sensitive to CO2, but also, of course, in general, phytoplankton may respond to increased CO2 because they are fueled by their photosynthesis fueled in the first place anyway. Right, let me see if I can move to the next slide. Yes, so we performed the meta-analysis, as I said. So we collected data. Well, I say we, but well, Karen did that. She was very active. She's very good at this, and she got almost like 3,000 results. Well, we also did it. I also did quite a few of those, actually, I realize now, because we screened all the titles, and then when we were unsure, we checked the abstracts, and we ended up with 144 studies. And out of that, we got nearly 170 different independent data sets. And then we could look at the response ratios. And in this case, I will show response ratios of CO2 and temperature of growth rate. So what we see here is, for instance, of different harmful algal bloom species, here a whole list. Here on the x-axis down here, you see the response ratio of growth rate of high CO2 over low CO2. So if this is positive, it means that high CO2 increased the growth rate. And if it's negative, high CO2 caused a decrease in growth rate. Well, first we see that there's a lot of data on dinoflagellates and not so much on the other groups, except for maybe some diatom species. So that's already a first interesting feature, right? So that gives us, so these meta-analyses typically give us also some idea on what is out there. What we also see is that there's quite some variation, but in general, dinoflagellate responds positively to CO2. So they increase the growth rate with CO2. So do the diatoms that we had in the studies, and the other studies we had actually too few to really say something about. I mean, there was a lot of variation because, well, very limited the number of studies. So there's an overall maybe positive effect on these hubs, but well, I'm inclined to say that we mostly see these diatoms and dinoflagellates. That's where we can say something about, right? So they increase their growth rate with response to CO2. Interestingly, we also had some data on non-harmful algal bloom species, so on other groups that did not include the harmful algal species. And there actually we did not see a response in any of the groups towards CO2. And I think that's quite intriguing, and it's still something we're not sure of what causes that difference. But well, I thought it's kind of pretty intriguing how that works. So definitely something we will investigate further. But overall, we see that harmful algal bloom species especially dinoflagellates and diatoms, respond to CO2 by increased growth. If we look then at the temperature effect, that's a little bit more complicated. So here we have again a response ratio to the background temperature, which is the temperature to where the species have been isolated. So at the time of isolation, right? And then this is the latitude where they have been isolated, and this is the absolute latitude, how we call that. So it's really, this is at the tropical, and then we go to the temperate regions, right? Both in the southern and northern hemisphere are in here, but it's mostly northern hemisphere data that we have. And what we see is that temperature response really depends on this. So if you're close to the tropics, then an increase in temperature generally has a negative effect or no effect at all. And then in the temperate regions, we actually see a positive effect of temperature with respect to growth rates. So the response to temperature really depends on where the species have been isolated and what the prevailing, the local temperature is. And this probably has to do with of course, the temperature optima. So the closer species are to their optima, and in the tropical regions, they are probably already close to their optima. Then if you increase temperature there, they may tip over this optimum and actually get negative responses. We also see that in a map. So here we see the locations of the different groups of species where they have been isolated. And here we have the maximum growth rate based on this temperature optimum. So what is the temperature optimum? And then the local sea surface temperature. So if this optimum is above the sea surface temperature, we get a positive value, right? And the higher above, the more positive value it is. And that's where we expect then also a greater potential of warming promoting growth of these species. Whereas if the species in the local conditions are already close or below their maximum, where the sea surface is already above that maximum, then of course we can expect negative effects. Well, that's also what we see here. So in the species, we see generally red in the temperate regions and blue here in the more subtropical, tropical regions. Here, another point that we see is that we miss a lot of data in the tropics in the first place, but especially also in the southern hemisphere. So this is definitely something that needs attention to get more data on and fill in more of those gaps there. So just to recap this part, the impacts of elevated PCO2 and temperature on growth of harmful algal species. We see higher growth rate, especially in temperate regions, for temperature, and of course CO2. We see higher growth rates in all the species. And there's potential of reallocation of energy and elements, at least for the dinoflagellates. So we had a whole chunk of dinoflagellates. And part of that response may, of course, be explained by CO2 fueling photosynthesis in general, but part may also be due to this reallocation from downregulation of CCM. So that's one of the lines we also want to follow up further. Then the other thing I wanted to highlight is a experiment. So this is impacts of elevated CO2 on hubs in the natural community. This experiment was performed in the Gulma Fjord. For those that do not know it, I never heard of it, I must say, but this is Sweden. Somehow Sweden is not given here, but anyway, Norway, Denmark, so you see it's right there near the Skagerrak. And this is where the Gulma Fjord is, and here are mesocosm bags. They are 17 meter deep and about 2 meter in diameter. And they are really long, deep bags, so to say, that are enclosed from the surrounding waters. And then they contain the community in early spring, and they were left in there until early summer. So this was a really long-term outdoor mesocosm experiment. And half of the system was exposed to high CO2, and the other half was just left open, so not exposed to changes in CO2 at all. And just to show that I was there, because actually I was only there for 1 week, because I was I finished my postdoc, and then someone else could actually work on the position we had for this. So I could only visit it for 1 week. This is proof that I was there, also for my colleagues that joined me, just to be sure that remember that I was there shortly. Right. But most of the work was done then by Silke and many of the other colleagues there as well. So what we expected was that with more CO2, we expected a higher harmful algal bloom biomass. Same reason as before. So we expected stimulated growth, and therefore also the potential for more biomass, also because there's more CO2 in the system. We also expected more toxins because of this biomass, not per se because more toxins in the cell, except for domic acid. So that's where we expected more toxins in the cell. So domic acid is this ASP. This causes this ASP that I showed before, and that's a carbon-based compound. And earlier studies have shown that the amount of domic acid that we see here on the y-axis in a cell can really increase with CO2 that we see here from white to light gray to dark gray. There's an increased CO2. There's a really dramatic increase in amount of domic acid production in the cells, especially under P-limited, well, actually only under P-limited conditions. So that's really important aspect of this. So if the systems would run into phosphorus limitation, then we expect to see an increase in domic acid per cell. And then, of course, depending on how the biomass changes, this may lead to an increase in overall toxin concentrations as well. So first we looked at the fjord itself. Because there was monitoring data, which is very important. So this was long-term monitoring data. And what we could see in, well, we analyzed that. I don't show all the data here, by the way, but this is the year where we sampled. So this is the period of the experiment. So this blue shaded area. And at the end of the season, we see that there is, in the fjord itself, an increase in Alexandrium, a certain Alexandrium species that are associated to an increase in a certain toxin, goniodomin. So that's at the end, but I will say more about that because actually that is not so important here. But what is more important is that we see also domic acid producers in the water. So here we see Pseudonychia seriata, a small peak here, and that there we at the same time see an increase in domic acid concentrations in the fjord water, right? So again, this is not in the mesocosms, this is in the fjord. We also see some other Pseudonychia later in the season, but there were no toxins observed. But here we do see toxins, right? So there is a toxic Pseudonychia in the fjord, and also a toxic dinoflagellate, Alexandrium, that's a dinoflagellate. If we look at the diatom abundance, this is the first results of the whole experimental time. So from this is the experimental time. Here we see in the line first. I just want to show what we see here. So this is the abundance. So in terms of chlorophyll concentrations, this is in the fjord. So we see dynamics, quite high dynamics in the start. But then here we see a peak in the fjord, a decline, and then later a small peak, and then decline, and then at the end another peak. And we call these different phases. And I want to focus on phase 2 and phase 3 here, because this peak in the fjord is associated, is at the same time we also see peaks in our mesocosms. And in blue, we see the ambient CO2 concentration with the variation. And in red, we see here, I hope you can see it, we see the high CO2. And for instance, in phase 2, we see that there's so much variation and there's hardly a difference between, well, there's no difference between the means at all. And that's of course partly because we work in a complex system, so high variation. Phase 3, we actually see that there is a higher amount of chlorophyll, and in another study we showed there's also higher primal production in the system, by mostly caused by diatoms because they're most abundant there, and that's where so we see more higher primal production and quite some diatoms there. If we look at dinoflagellates, we actually see, well, this was a little bit disappointing. We see some dinoflagellates at some point coming up in the fjord. So, well, that would be interesting to also study the effects of CO2 in the mesocosms. But actually in the mesocosms, we see some dinoflagellates that start, but then actually they just completely disappeared. And the reason for that is that we, of course, we close the systems, because it's some, you start the experiment, you close them. And dinoflagellates have a benthic phase. They produce cysts. And these cysts lay on the sediment. And at some point in time, mostly in spring, later spring, some even in summer, they come out of the sediment. And we knew that, of course. And for that reason, we also seeded the experiments with fjord water at a regular base. But yeah, we could not, of course, exchange too much of the mesocosm with fjord water, because then we would not have experiments. So yeah, this was a kind of a trade-off in the experimental system, and yeah, we did not include enough to really allow the dinoflagellates to develop in the mesocosms as they did in the fjords. So we don't have any data on the dinoflagellates, but we do have the diatoms, of course. Well, if we look at domic acid concentrations in the water that is associated to the particulate fraction, so to the cells, then we see that the CO2 effect is there. So we see an increase in domic acid concentrations associated to this biomass with more CO2. And that really is linked to biomass. So here we see, again, domic acid. This is a log domic acid concentration. Here we see diatom abundance. And overall, the higher the diatom abundance, the more domic acid we see, which makes sense. So these are really the toxic species. But it's time dependent, so we see some changes in this relationship over time. Then there are also other factors, so it's not only biomass, and this is also I put in for Paul just to have at least something of a ratio. So this is again domic acid. It's almost the same plot, but then we included in the model some different components to explain these toxins. So we could explain it again with the CO2 effect, but also we see that domic acid increases with the C/N ratios in the water, so the particulate organic carbon and particulate organic nitrogen ratios. So the higher C/N, the more of this domic acid, which stoichiometrically makes sense because these are really carbon-rich compounds. And also we see an increase with ammonium, and that's just more, that is linked then to the overall biomass. So ammonium fueling the biomass, and with that, the toxins that are produced and thus present in the water. So just to recap that part, so impacts of elevated PCO2 on hops in natural communities. So we see high variation with treatments. I mean, that's just something we have to deal with when we do natural experiments. Many people that do that are used to it. I had to get used to it. Mostly I work with these little laboratory-based systems. So for me, it was a big shock, but we could of course still find some, really see still some trends, and that was mostly because we really divided it only in two treatment, control and one treatment. So we had enough replicates, we had 5 replicates each, and that at least gave us the power to get some idea on effects of CO2, and we found then higher domic acid concentrations with more CO2. Important there, I have to note, is that the second bloom, so we only see this effect in the second bloom, and that's also where nutrients were low, and especially phosphate was low and presumably limiting. So we did not do bioassays to confirm that, but looking at the overall N/P ratios in the system really point towards the point that phosphate was limiting, and that of course matches then also with laboratory scale studies that I showed in my hypothesis. Right, so that's the recap of the 2 experiments. So what I hope to have shown here is just 2 examples in how we can approach this question of what are the impacts of global change on harmful algal blooms. So we can look at cellular levels and cellular level in lab-based studies, and we can expand that by doing meta-analysis approaches, right, to look to many more species and maybe also genotypes. And I have shown how this holds in how we can answer it when looking at natural communities, but also the difficulties that we get with that, of course. For an outlook, I just want to highlight a few points, because there's still this issue, of course, because these are examples how it works in one system, or maybe in laboratory experiments, and maybe it is around the world, but it's still laboratory experiments. So one of I think one of the challenges we still have is to continue developing mechanistic frameworks that allow us to scale from cells to elements. And also Paul works on that, like ecological stoichiometry is one of those frameworks that we can apply for that, and we can develop that further. Another aspect is how we deal with multi-stressor approaches, right? I already mentioned that some of the effects, like CO2, depends on nitrogen or phosphate availability. But of course, there are many more stressors out there, and that tells something that we can maybe test multiple stressors, but how do we still keep control of that? Because, well, you can only handle so many different treatments. So if we go multifactorial, how multi do we make that? Oh, that's not, sorry. I hope you can still see it. I pressed accidentally on end. And then the last point I want to highlight here is how can we go from an understanding? So let's say we get this understanding and we can test that in actual systems. How then can we go to predictions, right? So that we can really make at local areas and tell the communities also when you can expect certain blooms. And then maybe governments may respond to that by closing off certain waters, etc. So that's just some thoughts I have here as an outlook. That's some of the questions I'll be working on for the next years. Lastly, there's one more slide. I just want to highlight those interested in harmful algae. We will have a hybrid. Probably it will become a full virtual meeting given the chance for a third wave and everything. So probably it will be a virtual meeting, but it definitely will be from the 10th to the 15th of October. And so I do some advertisement here. So if you're interested, keep check on the website. We keep up in the next weeks. We will give some important updates also on the program, keynotes, and registrations. So with that, I want to finish and thank you all. Well, if you're still there, for your attention.
Thank you so much, Dr. Van de Waal. So now we will open up for questions. I encourage all audience members, if you do have a question, just unmute yourself and ask, or if you are not comfortable, feel free to write in the chat and we can kind of go from there. I do see 2 questions already, so we can start with those. So one of our faculty members, Dr. Xenopoulos, says, great talk. And she says, given that freshwaters are super saturated with carbon dioxide, I don't expect the same output for lake HABs. Can you comment?
Yes, shall I stop share, by the way? Is that nice or not? Because then I can maybe see everyone. Is that
Sure.
okay?
Yeah, go for it.
Oh yeah, that's better. Giving a talk, I actually don't see anyone. So now I actually see that there are people out there. Right. Yeah, freshwater systems are often super saturated with CO2. That's totally true. That's fueled by the dissolved organic carbon from runoff. And there, I don't think it's so much interesting to see the effect of elevated CO2 from the atmosphere, but it is still super interesting to study CO2 by itself, but then on a broader scale, because there we also see concentrations going up to 3000 ppm, but in summer it can go down to 50 ppm or so. So there we can just expand the axis. So in the natural systems, we may scale from 200 maybe to 800 ppm. That's might be realistic, but in freshwater systems, we can broaden that scale and still study then CO2. Another aspect related to that, by the way, is that we have very poor insights in the effects of pH and CO2, so the difference in that. So of course, if cells consume CO2, pH goes up as well. And there are only a few people that really study that during blooms, but pH can have a lot of direct effects as well on cells. And I think that is very poorly studied.
I have perhaps maybe a very naive question. So in your last slide about kind of future outlooks, you talk about going from or gaining a lot more insight through studies and then being able to use that to predict then, and where governments could use that information to predict. How does one go about, like, what is enough information? What needs to happen before you can get to a point of prediction?
Yeah, that's an excellent question. I think that is really, so there are different ways you can tackle that, right? So I think that's a study by itself. So how much do you need to know to make sensible predictions? I think for chlorophyll and biomass generally, we do quite well with rather simple models or general models with general parameters. But also that's why I showed these different components that determine bloom toxicity. I think there we can actually we still can make a step in adding some components to that chlorophyll. So what are the relationships with the biomass, for instance, and toxin levels if we add 1 or 2 species? So there are models, for instance, out there that already look at competition between major phytoplankton groups like cyanobacteria, diatoms. So we may be able to connect some information to those cyanobacteria. But you're right. I mean, it's how far do you go? Is it ever enough, especially for people working in a lab? I mean, I say that I understand things in the lab, but still there's a lot of things that we don't understand, of course. But I would say there's two ways. So using statistical models to look at phenomena that we had already. So that can already help a little bit to see, for instance, what are the boundary conditions where we see toxic blooms. So that can already give some kind of early warning, let's say salinity and temperature, and given these circumstances, there's a certain likelihood for a toxic bloom. That's one. And the other is then more mechanistic models. And yeah, there I think you can go very far. And there I'm not so sure of how far should you go.
Great, thanks. Yeah, so any other questions for Dr. Van de Waal?
I have a question.
Go ahead.
Yeah, thanks for that talk. That was really great to see all the different ways that you can do science. That was a really, really nice graphic of that. It's really kind of a trivial question, but you said side effects of mesocosms. Do you actually literally mean the sides of the mesocosms, or you're just talking about the fact that you didn't seed them properly or whatever?
Actually, both. Yeah, you're right. So it's mesocosms have, especially the indoor systems, we had a lot of side effects, but we tackled that by actually measuring it. So we took strips and we could measure the periphyton. The outdoor mesocosms were cleaned very regularly, but of course, it still has, of course, the side effect of the sides. But in general, I also mean side effect that you enclose a system, right? So it is really not the real system anymore, and we also see that in this Guma Fjord experiment. So we see that the blooms we still get at the same times, but we see much higher biomass, for instance, in the mesocosms. This is something very typical of mesocosm studies. So we cannot literally translate these studies to the field, but the trends, I think we can learn from those and see, and then of course test that again, maybe with monitoring data and real life data.
Thanks.
I'll chime in. So this is from the far west of the USA, former alumnus. I have a question. So I'm a limnologist as well. So I've done mesocosm experiments, and I'm just wondering, the surface area, the volume ratio is just phenomenal in 17-meter mesocosms. And Erica just alluded to that. And then you said you cleaned them. Were your mesocosms open to the bottom, or were they closed at the bottom?
They were closed at the bottom.
They were closed. So all the material that you're sloughing off actually accumulates on the bottom then, right?
Yeah, and that's then quantified. Yeah.
So you quantified it?
Yeah, that has been quantified. So it was a huge consortium, but so it funnels down, and then it was collected regularly, and yeah.
And then you obviously had a tidal compartment as well, right?
Sorry?
You had a tidal compartment on those mesocosms?
Like
the tide rising, coming and going with the water?
Yeah. No, of course, that was there were a lot of issues. I mean, it is a fjord. So actually the first issue was that it was first closed with, now I have to say it correctly, more saltier water. And then a fresh water surge came in. So then all the water went down. So suddenly the mesocosms were not like straight lines, but they became very narrow and then very like a ball. So yeah, I mean, I laugh about it now, but that was not good because of course some broke because of that as well. So that's really, really tough stuff. And this, I have to say, is these mesocosms, that's really huge operations. And it's a lot of effort from the Geomar group. So Ulf Riebersel coordinates those. And they have a lot of experience with that, but this was the first time for such a long study and also in the fjord. So that was definitely one of the issues. And you only close it at a certain time, right? So yeah, that is really a limitation then again. But yeah, that's a difficult thing.
Yeah, I wish you could do a whole seminar just on that, because the students here, like, that's a phenomenal undertaking to put those mesocosms in and actually run those experiments. Your one picture doesn't do it justice.
No, you can, and especially as I said, so Ulf Rubicel and his group, they have a lot of experience on these specific systems. So the indoor microcosm we use, that I already find complex enough. So, but you're right, and you're right. I mean that we always have to be careful about the systems that we use, right? And there's always going to be confounding factors. And I learned with our microcosms is then rather than to well, we accounted for it, so it's not really like it becomes a very small pond, so to say. But there's also a lot of surface area in that pond, right? And also, if you look at periphyton in plants, I mean, there's a lot of periphyton primary production in general in freshwater systems. If you also consider macrophytes there, and in that sense, it is not bad to have a component of that as well in the mesocosms. And by being able to monitor it, the photosynthesis, biomass buildup, composition, etc., you can at least also take that part into account. Yeah.
Great, thanks.
We have time for a few more questions if anyone else has one. All right. Well, I guess we can end there. Again, thank you so very much, Dr. Van de Waal, for giving us our seminar. We really appreciate it, and it was a very interesting talk. So